Hello students, welcome back to the channel. In our last video, we have given the discussion about the chemical reactions of individual amino acids that occurs due to the amine, that is NH2 group present in the skeletal structure of an amino acid. If you have not seen that video, the link has been given here and you can click on to this so that you can direct to that video. So in this video, we shall look over about the chemical reactions of the amino acids which is due to the another functional group which is present in the skeletal structure of an amino acid, that is COOH group. So today we shall look over the different kinds of reactions that a amino acid possesses, which is mainly because of COOH group. The first reaction that we shall look over is the reaction of amino acids with sodium hydroxide. Since we know that sodium hydroxide are basic in nature, since sodium hydroxide are very strong bases, COOH group which is acidic in nature, right? So when a base interacts with acid, obviously neutralization reaction will occur and that results in the formation of a salt. So like how we have seen in case of chemical reactions of amino acids due to NH2 group, when amino acid reacts with HCl, how it forms a salt. In a similar trend, when amino acid reacts with sodium hydroxide, that also results in the formation of salt. Strictly speaking, sodium salt of amino acid will be produced. Let us see the reaction pathway of this one. Let us consider the amino acid CHR NH2, and here it will be COOH, the amino acid, and this amino acid will be subjected to react with sodium hydroxide. So OH will be in the form of OH minus and sodium will be in the form of Na plus. This OH minus will pick this hydrogen, and so that results in the formation of elimination of H2O molecule, so that the bond pair of electrons present between oxygen and hydrogen will be donated to this sodium, so that results in the formation of sodium salt of that corresponding amino acid. And we know that R is the side chain, that is COONA will be the product. So this is the amino acid. Upon reacting with sodium hydroxide, we are going to get sodium salt of the corresponding amino acid. So this is the general reaction we can give. Let us consider one simple example. For the illustration, we shall consider alanine as the example. CH at the side chain, there will be CH3, and one functional group is NH2 and other one is carboxylic group, that is COOH. When you react this alanine with sodium hydroxide, this hydrogen will combine with this OH and that results in the elimination of H2O. So the bond pair of electron will be donated to this Na that develops a bond between oxygen and sodium. So the resulting product will be NH2 CH CH3 COONA. So this is the product, and the amino acid that we have considered over here is alanine, and we should know what is the name of this particular product which is formed during the reaction. Okay, so carboxylic acid is the first one. So 1, 2, 3, right? So there are 3 carbon atoms in the parent chain, and at second position we are having amine group. So we should call this as 2 amino, and 3 carbon atoms are there, and since it is in ester form, so 3 amino sodium propan, and the functional suffix for the ester is OH. So the complete name of the resulting product when alanine reacted with sodium hydroxide is 2 amino sodium propanoate, the simple one. Or else we can do one more example with another amino acid. This time let me consider glycine, CH2 NH2 COOH. This is the simplest amino acid molecule, and if you react the same with sodium hydroxide. As usual, the H will combine with this OH, and that results in the elimination of H2O. So finally you're going to get NH2, CH2, COO, Na, right? So this is the product produced when you react glycine with sodium hydroxide. So here 1, 2. So 2 carbon atoms are there. So at the second position we are having amino group. So 2 amino, 2 carbons are there, sodium, ethane, OH is the complete name of this product produced when glycine is neutralized with sodium hydroxide. So as a whole, what we can say, when amino acid is treated with a strong base such as sodium hydroxide, it results in the formation of sodium salt of the corresponding amino acid. Next, the second reaction of amino acids due to COOH group will be the reaction of amino acid with alcohol. COOH group, which is acidic in nature, and when you react the same with an alcohol in presence of a suitable acid, we can say that results in the elimination of water and to form a corresponding ester. Since if you react an amino acid with alcohol, it results in the formation of an ester. So we can call this reaction as esterification reaction of respectively amino acid. So esterification of amino acids. So now we shall see the mechanistic pathway for the reaction of amino acids with corresponding alcohol. So here first we shall consider an amino acid CH, R, NH2 and COOH, and we have to react this with an alcohol. So let me consider R dash OH. So this is the alcohol and this is the amino acid, and hope you are well aware, if we are performing an esterification reaction between a carboxylic acid and alcohol, the presence of an acid is mandatory, right? Provided the acid should act as a catalyst. And here in this case of esterification reaction of amino acid with alcohol, the catalyst which is going to be used is hydrochloric acid. So here you may get one more doubt, that in general for esterification reaction, the acid material which is going to be taken regularly is sulfuric acid. But here we are not going with sulfuric acid. The reason behind this is, if you use sulfuric acid or even if you go with the nitric acid, the reaction will takes place in such a way, instead of esterification reaction, the amino acid may undergo oxidative decarboxylation, and the resulting product will be completely different from the needy reaction. And along with that one, if you use sulfuric acid, which can act as a charring agent as well, the biomolecule may get charred, and to avoid all these phenomena, the usage of sulfuric acid and nitric acid will be avoided, and instead we will be considering HCl. And here the HCl will play multiple roles. The thing is, the HCl that what we have taken over here will act as catalyst, as well as it will react with amino group. One more doubt may comes to your mind, why it should react with amino group? Actually the reaction is supposed to happen between carboxylic acid and this alcohol. What is the necessity of reaction of HCl with amine group? This doubt may comes to you. The answer for the question is, here if you use alcohol, this alcohol may react with amine group also. Here also we have replaceable hydrogens, and this hydrogen may combine with OH group, and that results in the elimination of water, and the alkyl group may attach to the nitrogen atom, and that results in alkylation reaction of amino acid. But here our needy thing is, our expectation is esterification reaction. If alkylation is supposed to be prevented, this NH2 group is supposed to be blocked. Then only esterification will occur smoothly. To carry out this one, we use HCl, which is acting as both catalyst as well as will react with NH2. Okay. So in presence of HCl, amino acid will interact with alcohol and that finally results in the formation of NH2CH or COOH dash. That means the ester of amino acid will be produced. So this is simply called as esterification reaction. But here the detailed mechanism will explain you how actually the esterification reaction of the amino acid will occur. And now we shall proceed further to see the detailed mechanism of this esterification reaction of amino acid. Now let us see the mechanism. So firstly, the amino acid tend to react with the hydrochloric acid, COOH. So firstly, the H plus ion of the hydrochloric acid will comes into picture, and here the lone pair of electrons that are there on the nitrogen atom will be donated to this H plus, and that results in the attachment of this H plus to NH2 so as to form NH3 plus and CH or COOH. This will remain as it is. Okay, and to balance this positive charge, the Cl minus content of the HCl will also comes into picture so that we get the hydrochloride of the amino acid. Now the alcoholic content will comes into picture. So R dash OH. Okay, so here the thing what happens, OH will come from the carboxylic acid and H will be from the alcohol, and that results in the elimination of 1 unit of water, so that you are going to get NH3 plus Cl minus CH R, and here you will be having COOR dash. So hydrochloride of the ester of the amino acid has been obtained, and now in order to get the required ester we need to eliminate this HCl. So in order to perform that one, so what we are going to do here is we subject this ester to react with AgOH, silver hydroxide. So either we react with silver hydroxide or we will be reacting with moist silver oxide, that is Ag2O. Okay, so if you use AgOH, and here we will be having Ag plus and OH minus, the OH minus will pick the excess H plus, and that results in the elimination of H2O, and the Ag plus will pick the Cl minus, and that results in the elimination of AgCl, and finally you are going to get NH2 CH R COO R dash. That means the amino ester of the corresponding amino acid, right? So this is the ester of amino acid we are going to get when you react the amino acid with alcohol in presence of the mineral acid HCl. So now we shall look over into some simple examples for the esterification of amino acids. To illustrate the esterification reaction, here we have considered valine as an example, and let us react this with the simplest alcohol, that is methanol or methyl alcohol. When valine is reacted with methanol, as I said, the OH content will pick this H, and that results in the elimination of H2O in presence of HCl as well as in presence of AgOH. Okay, so in presence of HCl, initially this NH2 will pick the HCl so as to form the corresponding hydrochloride. Then the carboxylic end of the amino acid will react with the alcohol so as to form the corresponding intermediate. Once again, with reaction of AgOH, the final product will be obtained. Now we shall write the final product of the reaction: CH, CH3, CH3, CH, NH2, and here it will be COOCH3. This is the final product we obtained when valine is esterified with methanol. So now we shall give the name for this particular material. First we shall give the numbering 1, 2, 3, 4. So 4 carbon atoms are there in the parent chain, and at the second position we are having the amine, and at third position we are having methyl. So it will be 2 amino, 3 methyl, and 4 carbons are there in parental chain, and the esterified group is methyl butane OH will be the name of this material. So that's the simplest example we can give for the esterification reaction of the amino acid. So the next reaction will be the third reaction of amino acids due to COOH group is reduction of amino acids with the strong reducing agent such as lithium aluminum hydride. When amino acids are subjected to the process of reduction, so we know that if you have written like this, that indicates the nascent hydrogen, right? So when these amino acids are subjected for the reduction process in presence of a strong reducing agent such as lithium aluminum hydride, the resulting products will be the corresponding amino alcohols of the corresponding amino acids. So when amino acid is reduced, you are going to get amino alcohols. So now we shall see the simplest examples so that you will clearly understand what happens when an amino acid is subjected to reduction. Let us consider the same simple example, NH2 CH2, where I am going to get glycine. Here I am going to take glycine. When glycine is subjected to the process of reduction in presence of lithium aluminum hydride. Okay. So in presence of lithium aluminum hydride, 4 moles of nascent hydrogen will be utilized, and this NH2 will remain as it is, CH2, and this carboxylic acid will completely changes that to form the carbonyl group will changes into a methylene group, and this OH will remain as it is. So 1 unit of water molecule will get eliminated so as to form the corresponding amino alcohol. So here we have both amine group as well as alcoholic group. Let us give name for this molecule, the amino alcohol which is produced, that is glycine upon reduction will converts into, so 1, 2. So we are having this amino group at second position. So it is 2 amino, and there are 2 carbon atoms. So it is ethane, and it's an alcohol. So the suffix should be ol. So the name of the material produced after reduction of glycine is 2-amino ethanol. We can take one more example. So let us go with alanine, CH, CH3, NH2, and here it will be COOH. That is, we have taken alanine, and when it is subjected to the process of reduction, as I have told you, 4 moles of nascent hydrogen will be utilized in presence of lithium aluminum hydride. So 1 molecule of water will get eliminated, so that the product that is going to produce is NH2, CH, CH3, and once again carbonyl group will changes into CH2 and alcoholic group will remain. So the product will be named in this way. Once again, the second position is having amino group, so it will be 2 amino. 3 carbon atoms are there in the parent chain, so it is propanol. That's the product produced when alanine is reduced with lithium aluminum hydride. So these are the simplest illustrations through which we can explain the reduction of amino acids. In all those 3 reactions, we have noticed that the alpha COOH group of the amino acid tend to react with the reagent so as to produce the product. But here, the reaction of ammonia is very special. That means the COOH group which is going to react with this ammonia is not the alpha carboxylic group. The one should be the non-alpha carboxylic group. So we can get a point that this reaction is going to given by those two amino acids which are acidic amino acids, that mean which are having a carboxylic group in their side chain, that means aspartic acid and glutamic acid. These two amino acids will give this reaction. That means the amino acid containing non-alpha carboxylic group will react with the ammonia so as to produce the resulting product, and we should know which is the product is going to produce in this particular reaction. So let us consider the illustration. The amino acid which is considered over here is the acidic amino acid, that is aspartic acid. The aspartic acid contains two COOH groups. One is the alpha COOH group and other one is non-alpha COOH group. Now the reaction is due to this non-alpha COOH group, and this aspartic acid is reacted with ammonia. Here the lone pair of electrons present on the oxygen atom will be donated to the hydrogen atom. So the bond will develop between this OH and H, and so that to balance the lone pair of electron, these bond pairs will shift on to oxygen. So the bond will get break between carbon and oxygen, and this carbon will suffers with electron deficiency. To satisfy this electron deficiency, the lone pair of electrons present on the nitrogen will be donated to the carbon, so they develop a bond between carbon and nitrogen. Again, to balance the lone pairs, these two bond pairs will once again shift on to nitrogen, so that the bond will get break between nitrogen and hydrogen. So formation of one new bond, breaking of two older bonds, and that finally results in the formation of CH, NH2, COOH is the skeletal structure, CH2, C double bond O, and this side we will be having NH2. So a carboxylic acid has been converted into a acid amide, and we know that this is the structure of aspargine, means one amino acid has been converted into another amino acid. This is the reaction how actually happens between aspartic acid and ammonia. The same kind we can observe with glutamic acid. When it is reacted with ammonia, that results in the formation of glutamin. Okay, so glutamic acid, which upon reaction with ammonia will results in the formation of the next acid amide, that is glutamin. So this reaction is very important in the field of living system. The reason behind this is, in our body, due to the biological reactions, the ammonia production will happen. But ammonia, if it is remains in the body, that creates more toxic reactions in the system. So that what we call as ammonia toxification. To avoid all those ammonia toxification, by nature, the ammonia will tend to react with aspartic acid or glutamic acid, and that will result in the formation of the other amino acids such as aspargine and glutamin, which will be useful in the formation of protein molecule. So that in this way the reaction is so much useful. This reaction of amino acids due to COOH group is the most important one, that is decarboxylation of amino acids. We know that the meaning of decarboxylation is simply the removal of carbon dioxide. If you remove the carbon dioxide from a carboxylic acid, that process is actually called as decarboxylation. In order to perform the decarboxylation of any amino acid, we used to consider the sodium carbonate, or we will be going with soda lime, that is CaO plus NaOH. We will be going with the soda lime, that is CaO, that is quicklime, and sodium hydroxide. But here we are going to perform the decarboxylation reaction of a biomolecule, that is amino acid. So no need to, or it is not possible to go with the conventional decarboxyling agents, or we cannot heat much strongly to cause the decarboxylation. For that reason, we will be going with a specific enzyme materials, and the enzyme that we will consider for performing decarboxylation reaction is simply called as DPLP. So we should know what is this DPLP. DPLP is the enzyme called as decarboxylase pyridoxal phosphate. Pyridoxal phosphate is the full name of this enzyme, DPLP. And this enzyme is so much employed in performing decarboxylation reaction of amino acid. And one thing we are supposed to remember, should understand where actually this decarboxylation reaction will occur. The decarboxylation of amino acid majorly occurs in the main part of the cell, that is the energy factory, mitochondria. In mitochondria of the cell, the decarboxylation reaction of amino acids will occur. And now we shall consider one or two simple examples to show the decarboxylation reaction of amino acids. Here we have taken the aromatic amino acid, that is tyrosine, to show the decarboxylation reaction. So to perform the decarboxylation of this tyrosine, we will be taking tyrosine decarboxylase, a specific amino acid, a specific enzyme that will perform the decarboxylation reaction of tyrosine, or you can directly take DPLP, that is decarboxylase pyridoxal phosphate. So what happens now? Now this carbon dioxide, COO, this whole thing will eliminate. The reason is this bond pairs will shift here, so the bond will break between oxygen and hydrogen. Now this hydrogen is in need of electrons, so the bond pair of electrons will be donated to this hydrogen, so CH will become CH2. Let us see the final product which will be produced, NH2 CH, and here by receiving one hydrogen it will become CH2. We have one more CH2 and a benzene ring which is further attached with OH. So this is the product we are going to get when tyrosine is subjected to decarboxylation, that is elimination of carbon dioxide. The product produced after decarboxylation of tyrosine is an amine which is called as tyramine, is the name of this amine. So like how we got tyramine from tyrosine, similarly if you subject one more amino acid, that is histidine, for the process of decarboxylation, the product which is going to produce will be histamine. So histamines are the most important components regarding which we shall discuss in the later videos. Okay, so similarly if you react one more aromatic amino acid, that is phenylalanine, so whose structure almost resembles with tyrosine, only exception is OH will not be there in case of phenylalanine. So the name of the material will be, if you forget this OH, you will be having this phenyl, and the remaining thing is ethanamine, right? So the resulting product is called phenylethanamine, is the product you will be getting when this phenylalanine is subjected to decarboxylation. Clear? That means when you subject the amino acids for the process of decarboxylation, we are going to get corresponding amines after elimination of carbon dioxide. So these are the reactions of amino acids due to COOH group. So that's all for the session. In our next video, we shall continue our discussion of the amino acids and proteins with the topic of peptides. Thank you.